ഹലോ ഫ്രണ്ട്സ്, എക്സാമിങ് എത്തിക്കഴിഞ്ഞു. യു ക്യാൻ റിവൈസ് ദ ടോപ്പിക് digestion of protein and absorption. Welcome to Edge Learning platform. If you have not subscribed this channel, please do subscribe. You can press the bell icon and enable the all button. There are a lot of videos we can available for different types of exam, so please be supportive. Now we can study digestion of protein and absorption. Digestion. Dietary protein are hydrolyzed to amino acid by proteolytic enzyme, which can be easily absorbed. Dietary protein are hydrolyzed to amino acid by proteolytic enzyme, which can be easily absorbed. Proteolytic responsible for degrading protein produced by three different organs: stomach, pancreas, small intestine. What do you mean by digestion? Dietary protein are hydrolyzed to amino acid by proteolytic enzyme. Proteolytic responsible for degrading protein produced by three different organs: stomach, pancreas, small intestine. Digestion of protein, which include an important thing, is peptide bonds. Peptide bonds. Then characteristics of enzymes: endopeptidase and exopeptidase is there. Characteristics: there are two enzymes. First one is endopeptidase and second one is exopeptidase. And what is the speciality of endopeptidase and exopeptidase and proenzymes? Act on peptide bonds inside protein molecule. Example: trypsin, chymotrypsin and elasticides. Endopeptidase acts on peptide bond inside protein molecule. Example: trypsin, chymotrypsin, elastase. Exopeptidase means it acts at peptide bond at the amino terminal or carboxy terminal end of protein. Example: carboxy peptidase, amino peptidase at peptide bond at the amino terminal or carboxy terminal end of protein. Example: carboxy peptidase, amino peptidase. And zymogen, secreted as inactive zymogen, they are converted active form in the intestinal lumen. Proenzymes, the another name for proenzyme is zymogen. Characteristics of proteolytic enzymes. Proenzymes is another name is zymogen. What are the characteristics? First of all, we can study what are the characteristics of proteolytic enzyme. Endopeptidase acts inside the core of protein, forms small peptide fragments. Exopeptidase act from the amino terminal or carboxyl terminal end of protein. Aminopeptidase and carboxy peptidase. Prozymogens or zymogens, peptidases are secreted in the inactive form When zymogens reach the site of active and when zymogen reach the site of action, they are activated. They are activated. A small picture of digestion of protein. First of all, salivary gland, salivary juice, and salivary gland. Which type of juice is available? Salivary juice. Stomach, gastric juice. Intestine, intestinal juice. And liver, it is hepatic juice. Pancreas is pancreatic juice. Digestion in stomach. Digestion of protein starts in stomach. When protein enters the stomach, it stimulates the secretion of hormone gastrin from gastric mucosal cell. This gastrin in turn stimulates the release of gastric juice, which contain hydrochloric acid, pepsinogen, renin in infants. The pH of gastric juice is 1.5 to 2.5. First of all, there is no enzymes present in the mouth, so protein digestion takes place in the stomach. When protein enters the stomach, it stimulates the secretion of hormone gastrin from gastric mucosal cells. This gastrin in turn stimulates the release of gastric juice, which contain hydrochloric acid, pepsinogen, renin. The pH of gastric juice is 1.5 to 2.5. Digestion of protein begins in the stomach. We already know that. And stomach secrete the strong acid is HCl, enzyme pepsinogen. And what is the duty of HCl? HCl, which denatures protein, decrease the pH to 2 to 3, activates pepsinogen, kill some bacteria, and help in the absorption of vitamin B12. And what are the functions of pepsinogen? Pepsinogen secreted by chief cells and activated to pepsin by HCl, an autocatalyst. So from pepsinogen, with the help of HCl, it transported to pepsin. And pepsinogen is an autocatalyst and do pepsin. What is the function of pepsin? Pepsin adds on peptide bond formed by carboxyl group of peptide bond formed by the carboxyl group of phenylalanine, triosine, tryptophan, methionine. Phenylalanine, triosine, tryptophan. Tryptophan, it is an excellent amino acid group present in the milk. Methionine, it is methionine, the legume deficient protein. And peptide bond formed by carboxyl group of phenylalanine, triosine, tryptophan, methionine. And pepsin add-on converts protein into protease and peptones. This pepsin, which convert protein into proteases and peptones. Renin. Renin is what is speciality of renin? Renin, also called chymosin, active in infants, and renin, which help the curdling of milk, absent in adult stomach. Renin, which help in curdling of milk, and it is absent in adult stomach. Renin convert casein of milk into paracasein. Renin convert casein of milk into paracasein. It combines with the calcium to form calcium paracaseinate. It combines with the calcium to form calcium paracaseinate. Digestion of protein in the intestine by pancreatic enzymes. After stomach, as the acidic stomach contents pass into duodenum, the low pH triggers the secretion of hormones. Secretin stimulate pancreas to secrete bicarbonate. It neutralizes HCl and rises the pH from 1.5 to 2.5 to 7.0. Cholecystokinin stimulates secretion of pancreatic endopeptidase. What all included in the endopeptidase? Trypsin, chymotrypsin, elastase, exopeptidase, carboxypeptidases, aminopeptidases. Endopeptidase cleave internal peptide bond and result into formation of smaller peptide. An endopeptidase secreted by pancreas. Endopeptides secreted by pancreas are trypsin, chymotrypsin, and elastase. Trypsin is tryptophan, chymotrypsin is chymotrypsin, elastase is proteolitase. Aminopeptidase and di and tripeptidase means what? Aminopeptidase means present on luminous surface of the intestinal mucosa. It is an exopeptidase. Adds on N-terminal peptide bond, release free amino acid. Di and tripeptidases are present on the surface of intestinal mucosa. Act on dipeptide and release free amino acids. Enterocytes take up di and tripeptide. These particles are hydrolyzed to amino acid by intracellular dipeptidase. Exopeptidase cleave amino acid at the time from either the carbon or N-terminal of the polypeptide. Exopeptidase secreted by pancreas. Carboxypeptide, otherwise known as procarboxypeptide, aminopeptidase. What is the function of exopeptidase? It cleaves off amino acid at the time from either the carbon or nitrogen terminal of the polypeptide. What is the special functions of pancreatic peptidases? Secreted as proenzymes, endopeptidase, exopeptidases. Endopeptidase, which include trypsin, chymotrypsin, and elastase, and exopeptidase is carboxypeptidase A and carboxypeptidase B. Digestion of protein in the intestine by intestinal enzymes. The digestion of product of hydrolysis by pepsin, trypsin, elastase, chymotrypsin, carboxypeptidase is completed by the intestinal peptidases secreted by the mucosa of small intestine. Some of these peptidases are amino peptidases and dipeptidases. An overview of the digestion of protein. So at least we have to remember this much point. In mouth there is no enzymes. In stomach its pepsin and renin is present, and at the case of pancreas, trypsin, chymotrypsin, elastase, carboxypeptidase B, and in the intestine is aminopeptidase and dipeptidase. So this is a picture lumen cell, lumen present in the small intestine. Absorption of amino acid. Absorption of most of the amino acid take place by active transport mechanism. Absorption of most of the amino acid take place by active transport mechanism by Na plus dependent active transport system. Na plus amino acid is an active, it's a co-transport as energy requiring process. Absorption of most of the amino acid take place by active transport mechanism by Na plus present in the Na plus dependent active transport system and Na plus amino acid co-transfer. It attacks an energy requiring process. Thank you for watching. Keep watching Edjo Learning Platform for more exam tips. So just you study the important point, what all enzymes are present in each organ of the body in order to digest protein. Thank you.